The overall goal of this experiment is to measure microtubule dynamic instability in docetaxel-resistant breast cancer cells.
This method can help answer key questions in the field of cell biology about microtubule dynamic instability under physiological conditions and following various drug treatments. The main advantages of this technique are that it provides a simple, reliable, and physiologically applicable protocol for studying microtubule dynamics in living cells.
Before beginning the procedure, grow the breast cancer cells in complete culture medium in 10 cm culture dishes at 37 degrees Celsius and 5% carbon dioxide to the appropriate degree of confluency. The day before the experiment, rinse 24 mm poly-L-lysine coated glass cover slips with 70% alcohol and expose the slips to UV light overnight. The next morning, place one cover slip into each well of a 6-well plate. Then wash the cultures with PBS and detach the cells in each dish with 0.5 milliliters of trypsin EDTA. When the cells have begun to lift from the plate bottom, neutralize the trypsin with 5 milliliters of fresh culture medium and count the cells. Then add approximately 1 × 10⁵ cells to each cover slip, shake the plate gently, and return the cells to the incubator for at least 36 hours. To assay the microtubule dynamics of the cells, mix GFP-tagged alpha-tubulin stock solution several times by inversion to ensure a homogeneous solution without vortexing. Then replace the culture medium.